Good evening, everybody. Thank you for coming. My name is Tony Farrell. I am president of SEB, and it is my absolute great honor to welcome you to the 2014 BidderLecture. This lecture recognizes an outstanding contribution to a senior scientist in experimental biology. And it's a special thrill for me to introduce the person who happens to be my boss. We didn't fix this in any way. I have nothing to do with the selection of the BidderLecture. But my boss, Bill Milsom, is from UBC, absolutely amazing guy, and it's wonderful for me to do it. Bill is an exceptional person. He's a person who's either liked or loved. There's no other gray zone there. Everyone he contacts, he engenders this liking or loving. He's been an absolutely amazingly effective administrator. And how can you be an administrator and still be liked and loved? That's a stunning achievement in itself. Not only that, he's this incredible mentor. We saw letters from his students in a retirement dinner we had in Edinburgh called the Milsom Fest. And you wouldn't believe what people had to say about him. And he's been awarded at UBC for his mentorship skills. But tonight we're here to recognize an amazing scientist because he is indeed that. Bill is regarded by most in the field of comparative respiratory physiology as the guru. He's the guy you go to when you want to understand the big picture. And when you look at what Bill works on, he works on the big picture. He can pick up nerves. He's got hands of steel. He isolates isolated nerves. And then he'll talk about flying barheaded geese over the Himalayas. Now, if that isn't big picture science, I don't know what is. In addition, he covers a range of animals which is just truly impressive. It's not just barheaded geese, it's reptilians and amphibians and fishes. He loves vertebrates. In fact, he loves all animals, and I suspect we may see some of that tonight. So please welcome Bill Milsom, the BidderLecture for 2014.
Thanks, Tony. Well, thank you, and let me begin by both thanking you for coming and thanking the Society and those that nominated me for this award. It really is a prestigious award, and I really am simply delighted to be the recipient of it for this year. Now, I thought I'd take after Chris Wood gave this to me after his BidderLecture, a picture of George Parker Bidder himself, who was a marine biologist and once upon a time the president of the Marine Biological Association of the UK. And if you'll notice the years that are there, the amazing thing is George Bidder was the son of George Bidder, who was the son of George Bidder. They were 3 generations with the same name. So it gets a little bit confusing. But he's of the same era as 2 of my great heroes, and that were Claude Bernard and August Crowe. And these really were the beginning pioneers of comparative physiology that recognized that animal species could be used as a tool for unraveling problems. And in my own lab, I follow very much in their footsteps, using animal species as a tool, but also phylogeny and ontogeny as tools. And it's a bit of a motherhood statement here, but really the 2 key things that I focus on are looking at the evolutionary origins of physiological processes, how processes have evolved, and then looking at the plasticity in them and the way in which they can be adapted to allow animals to live in extreme environments. Of course, the bottom 2 are to put comparative physiology in the realm of biology in general, and that is that comparative physiology defines functional genomics at the whole animal level and determines the basis of biodiversity. Animals live where they do because they can because of their physiology. Now, this is certainly a overstatement, but my own work is a very, very small piece of that, and what it really focuses on is the gas transport cascade. What are the adaptations that match O2 supply with O2 demand and O2 elimination with O2, or CO2 elimination with CO2 production? And the process is a cascade that consists of multiple steps coming down from ventilation of either lungs or gills, depending on the species, and I'm dealing only with vertebrates here tonight. Then diffusion from air into blood, perfusion, the two convective steps to deliver that to the capillaries where diffusion into the tissues takes place to allow cellular metabolism. Now, my own work takes and looks at all the steps in that cascade, analyzes things from the cell level right up to the organismal and population level, and dwells on anatomy, physiology, but most of all, control. I am a control freak. That really is where my heart is. How are these processes controlled? And so most of my work is on ventilatory control at the organ system and organism level, although the work in the lab falls all over this, the grid that is shown here. Now, given that I do work on control, unfortunately, I'll begin with a control diagram showing a feedback loop. And I'm going to spend a little bit of time, probably the first 2/3, wandering through bits and pieces of this and talking about the evolution of these processes. We need to have a rhythm generator. We know that breathing is rhythmic, it is autonomic, and there is a rhythm generator somewhere that interacts with descending inputs from the brain, feedback from chemoreceptors and mechanoreceptors to produce a breathing pattern. And that pattern leads to ventilation, which, depending on metabolic rate, will determine the levels of blood gases, being modified a little bit by cardiac shunt, which again will then feed back onto the chemoreceptors and feed back to the central controller. So basic fundamental negative feedback loop. Now let's begin and take a little bit of a look at some of the evolutionary trends that we see in central rhythm generation. Central rhythm was sort of the holy grail of respiratory physiologists for over a century. The site is now reasonably well known, although there's still lots of argument. And I'm going to take you through a tiny piece of that here tonight. Put it in context, I want to take a look at a few phylogenetic trends. And these are the trends in the respiratory pumps, the muscles that drive those pumps, and the innervation that they receive. And really, when we start back with the hagfish and lampreys and come down through the fish and into the amphibians, the pump itself is a buccal pump. That is, it's the floor of the mouth that primarily the pump that's drawing in air and pushing it into the lung or over the gills. It's driven by branchiomeric muscles, muscles that are derived from the branchial pouches, and they're innervated by cranial nerves coming directly off the primarily the medulla and the pons, and these are the 5th and the 7th, 10th and 12th nerves primarily. As we come down to the air-breathing so-called higher vertebrates, we now have the buccal pump giving way to a suction or aspiration pump, and this is driven by axial muscles, primarily the muscles of the chest wall. They are innervated by spinal nerves, and those spinal nerves run up the spinal column to an area called the ventral respiratory group, and this is where the motor output for these pumps originate. So we see some definite evolutionary trends, both in the pumps, the muscles, and the innervation. And it gives rise to a series of questions. Where are the rhythm-generating circuits, the holy grail that respiratory biologists have sought? And what are the implications of what we see here from a comparative perspective on those pumps and their control, and should we expect to see homologies? Well, let me give you a little bit of background. We know that early in development, and here we see a scanning EM of a developing embryo, and you can see the developing gill pouches. This is actually a mammalian embryo. You can see the developing gill pouches, and we know now there's very good evidence that there are rhythm-generating circuits throughout the developing nervous system. They're associated with every pair of rhombomeres. There are 7 rhombomeres that make up the medulla. And with every pair, there is a motor nerve output, and these are primarily 5, 7, and 10. 12 also is a motor output associated with breathing. And there's a rhythm-generating circuit associated with each one early in development. There's also a rhythm-generating circuit associated with every spinal segment along the spinal cord early in development. As the organism matures, many of these rhythm-generating circuits lose their ability and disappear. Some are retained. We also know that rhythmic activities are very common and not just breathing. These include chewing, swallowing, suckling, vomiting, breathing, and locomotion. And so the circuits are there. The basis for these rhythms are there. And notice that many of these rhythms use the same muscles. And we know that the rhythm generators are in association with the branchiomeric muscles, the ones that come down between the developing gill arches, as well as the hypoglossal ones that come down behind and run up through the floor of the buccal cavity and form the tongue and associated structures at the bottom of the buccal cavity. So with that as a bit of a background, I'll run you through a little bit of the data taken from the literature as well as from our own studies. In the lampreys and hagfish, the data to date indicate that using an isolated brainstem spinal cord taken from the animal, so you've got the neural circuit in isolation with no feedback, no descending input, nothing to confuse the picture, that breathing is generated by a pair of rhythm generators, one on either side, associated with the 5th cranial nerve, with the trigeminal. So it's in a paratrigeminal location. There also tends to be another pair sitting back near nerves 9 and 10, but the primary one that drives breathing in this group is associated with the 5th nerve. As we move up into the fishes, and I'll lump in here chondrichthyes as well as osteichthyes, so far, and there hasn't been a lot of recent work, but the early work could not separate out a single site, but found a very dispersed series of neurons with respiratory rhythm that ran all the way from the 5th nerve back to the tail end of the 10th. Some studies suggested that the primary accumulation was near the 7th cranial nerve, and that becomes important a little bit later. Now, even though they couldn't separate out a single site, we know there are multiple rhythm generators in there because under certain circumstances, we're able to separate out a buccal rhythm that's separate from the opercular rhythm. So the buccal one is producing the major force pushing water over the gills, but the opercular flare in and out, drawing water over the gills as well. And here you can see, especially on the bottom, recording impedance from the buccal cavity as well as opercular pressure, that with an injection of cyanide, a mimic of hypoxia, had no effect on the opercular rhythm, but had a tremendous effect on the buccal rhythm. So the suggestion is that there has to be more than one rhythm generator associated with the production of this movement, even though typically the two of them are entrained and work together. In amphibia, there's evidence for 2 sites as well. Again, one of them in the region of the 7th cranial nerve and one in the region of the 9th and 10th cranial nerves. And in frogs, if you think about their breathing, there are these buccal oscillations that move air in and out of the mouth. And then there are the bigger contractions that lead to lung ventilation and inflate the lungs. And the buccal oscillator tends to be the one at the back, and the lung oscillator tends to be the one at the front. So again, we see at least 2 rhythm generators associated with movements in all of these animals, and that tends to be the case in mammals as well. Here we're looking at a section of the rostral part of the medulla and the pons, the spinal cord's back here, and the thinking bits are all up here. And the primary area for producing inspiratory rhythm is this pink area called the pre-Botzinger complex. And there's another rhythm generating site sitting right under the 7th cranial nerve called the parafacial respiratory group, which controls active expiration. We know in most mammals, us included, at rest, inspiration is active and expiration is passive. And early in development, it's the perifacial group that dominates, but through embryonic development, the Prebot-Singer complex takes over the predominant role and controls active expiration, which is what we see. So now if I put those pieces together, what we see then is these rhombomerit pairs, each of which give rise to an oscillator, the fifth nerve, perifacial and perivagal, and that in lampreys, all of them produce buccal oscillation, and this it seems to be true, of course, in the osteichthyes as well. We move up into the amphibians, and the perifacial takes on the predominant role, driving lung ventilation. Of course, back in the lamprey, it was the one in the paratrigeminal region which dominated. When we come up into reptiles, we have, again, oscillators in both regions, but the one that dominates, lung deflation always precedes inflation in a reptile, and this is the dominant generator. And then when we move up into mammals, expiration becomes passive, and the perivagal oscillator becomes the dominant one. So what we see then in terms of lessons learned from this is a caudal shift in the dominant site of rhythm generation associated with the change from a buccal pump to a aspiration pump, which is intriguing. Now, the questions that are left unanswered, partially answered, are what are the mechanisms that produce the rhythm at each of these sites? Are they the same at each site? And are they the same across species? And I'm going to show you some of the evidence that exists, but I'll point out that none of these questions are answered at this point. And in fact, through my talk, I'm going to talk a little bit about the questions that I've started with, the lessons that I've learned. Others may not have, and they may have learned very different things, and I may be wrong. And I'll talk a little bit about the unanswered questions, because these are the areas for graduate students to grab hold of and run with. Now, the next two slides are taken from a review article by Diethelm Rector and Jeff Smith, and they're a little confusing, and they're really just for demonstration purposes. But within the rhythm-generating sites within the brainstem, we can find neurons with very different discharge characteristics. Down at the bottom is the phrenic burst, which is going to move the diaphragm in a mammal. So when you see this burst, that's inspiration, and then we've got an expiratory phase that's broken into two parts, and I won't go into why. But note that some of the cells burst in expiration, some in inspiration, some are early, some are late, some are continuous, some are augmenting. So there are very different cell types, and we do know that the cell types are different at different sites, at least in mammals. They appear to be phylogenetically conserved in adult mammals, and the rhythm is produced due to the interaction of this network of different types of neurons. Now, what gives those neurons these characteristics are the channels that they have, and this is something I'm not going to spend any time on. I'll put this up from the review, and I just want to point out one ion channel in particular, and that's this one, which is a persistent sodium channel. And this channel gives the cells that contain it pacemaker-like properties. And one of the big questions always was, is respiratory rhythm like cardiac rhythm driven by pacemakers, or is it due to the network and network interactions? And when the pacemaker cells were found, people were excited, as they should be, but it turns out that in adults, these pacemaker cells are not essential. You can destroy and ablate them, and if you do, the rhythm still continues. It looks like in adults, their role is to stabilize the rhythm, but they are not obligatory for producing it. But one of the interesting things to us was a suggestion early on that they might be more essential in the embryo when the network is first developing, so that the pacemaker cells set up this intrinsic rhythm while the network develops, and then as the network becomes established, they take a secondary role, and now they have a stabilizing function. And the reason this was so intriguing to us was because we have a hypothesis we developed in the lab some years ago. that hibernation is due to the retention of neotenic or neonatal characteristics. So it's the hibernator is neonate hypothesis. And what this is based on is now a list of about 17 or 18 characteristics that adult hibernators share with neonates of heterotherms that do not hibernate, but that the non-hibernators lose during development. And these include things like the use of brown fat, a heavier reliance on white fat for fuel, left shift in the oxygen equilibrium curve, the role of the vagus nerve. And the list is quite extensive, but it also includes hypoxia tolerance and cold tolerance. And so one of our thoughts was that the pacemaker properties are retained in these animals, and it allows them, particularly with cold tolerance where there are long periods of apnea, where breathing does not occur, to start, stop, restart breathing. And this is something I'll show you a little bit more about later. So the preparations that we've used to address a lot of these questions, and as Tony alluded to, my lab is a little schizophrenic, but on the respiratory neurobiology side, we tend to use an isolated brainstem, so we have the medulla and various parts of the spinal cord. And in isolation, perfused with the appropriate solutions, the brainstem continues to produce a respiratory rhythm. C4 would be the phrenic nerve, and you can see phrenic burst discharge, and you can see the tongue, the upper airway muscles, and here the intercostal muscles, as well as the abdominal muscles, all firing, showing a typical respiratory rhythm with the brainstem completely in isolation. And this allows us to look at characteristics that are intrinsic to the brainstem with none of the confounding factors of feedback. Now, riluzole is a dirty drug, but amongst the things it does is it blocks persistent sodium current. And so our hypothesis was that if the neonates and the heterotherms were more dependent on pacemakers, then blocking pacemaker rhythm should have a more profound effect on their breathing pattern than it would in a heterotherm. And it's more or less what we see. So here we see that as we give increasing doses of riluzole, that we find that in older rats, so weaned rats that are more mature, it has very little effect, suggesting pacemakers aren't important. We see in the heterotherm that the breathing drops off very, very rapidly, and that the young rats are somewhere in between. So again, the data are consistent, but not totally conclusive. So at this point, coming back again to the questions, there are a fair number of models now developed on the discharge characteristics of the different neurons. So the cellular mechanisms are close to being unraveled. There's not complete agreement on the ways in which they interact. They are not homologous at each site, and it would certainly appear that they're not the same in adults of all species, although they may be during early embryonic development. And we have very little data to compare in non-mammalian species at this point. Okay, coming back to our diagram, I'd like to take a look now at central pattern generation as opposed to rhythm generation. And pattern now then is the manner in which that rhythm interacts with inputs from other sources, primarily chemoreceptors, mechanoreceptors, and descending influences from higher up in the brain. And let me try to demonstrate the concept of pattern as opposed to rhythm. Many animals, particularly mammals, but reptiles, amphibians, fish, have a nice rhythmic breathing pattern at rest. And it's led to the production of models that will produce a nice sinusoidal oscillation and a nice rhythm. But a number of years ago, a variety of comparative physiologists, and notably Nigel West actually, realized that many species have very simple lungs. And here we have a frog, a turtle, and in this case, an air-breathing fish. This is Protopterus, the lungfish. And they realized that what you could do is you could cannulate the tips of the two lungs and then tie off the glottis. And you could now artificially unidirectionally ventilate the animal, putting air in the tip of one lung. It will go through, come out the other lung, and you could measure the respiratory gases We could use an extracorporeal loop to measure what was happening in the blood. And you could now ventilate the lung so that oxygen was held high, CO2 was held low, and when you did that, the animals don't breathe. So that the rhythm is conditional, it needs some sort of an input, and it turns out the input it needs can be tonic. Doesn't have to oscillate, just a constant input to bring that whole network up above threshold and allow the rhythm to be expressed. And so these early studies said that breathing is conditional on an excitatory input of some sort under resting conditions. And we now know this to be true of all vertebrates. This is some work that Angelina Fong did in my lab, which really was a tour de force. Getting at embryos and working with embryos is difficult, but as we know, marsupials give birth at very, very early stages. And this is the young of a dunnart, which is a marsupial rodent. This is an Australian coin about the size of a Canadian or an American penny. And I'm not sure what that would be in the UK, but a very small coin. And you can see that the newborn is tiny. It's the size of a grain of rice. And Angelina managed to take the brainstem and spinal cord out of that and still record from the neurons. Now, when the joey leaves the pouch on day 0, there's no breathing. It's all cutaneous. There's no need to take a breath. And in fact, Peter Farrell has shown that at this point, the lungs are not connected to the outside. There's no bronchus, there's no trachea. They develop at about days 2 to 3. So the lungs aren't even connected. And you can see at day 5, you're only starting to get occasional breaths. And it's really not until 12 days post-natal that you begin to see continuous breathing. And what Angelina showed is that with this in vitro prep, that the rhythm generators are capable of functioning even at the day that the joeys emerge and would respond to CO2 by increasing their discharge rates and increasing the fictive breathing rhythm. So that the rhythm generator is ready to go at day 0, but it's not recruited until it's needed, which is between days 5 and day 12. So that again, the central rhythm can be generated, but is conditional and is not expressed until there is some other input. Now, when it is expressed, one of the things that's intrigued me for much of my career is that in some species, the breathing is regular, and here we have increasing levels of drive as we go from top to bottom. And this is a trace I forgot to put the reference on that I stole from Tobias Wang and Nini Skovgaard of a reptile breathing with evenly spaced breaths. But on the right, we see a bullfrog, and here we see those buccal oscillations. We see nothing when there's very, very low drive. We see the buccal oscillations come in with single breaths, and as the drive goes up, they come in pairs, and then they come in small clusters, larger clusters, and become continuous. And so, even under conditions, now I should point out that in each of these stages, the animals are being unidirectionally ventilated, so blood gases and lung gases are not oscillating. They're continuous and flat. So the CO2 levels, which are what are increasing here, are being held constant. Yet despite constant input, the breathing is episodic. Now these episodic patterns are common. And over the years, we've worked with many, many species that exhibit them. So here you see in fish, carp show this pattern as well. Amphibians, I've just shown you. Turtles, it's a very common pattern. We see it early in development in chicks as well as in newborn mammals. And we see it in adults, particularly of many of the hibernating species. Gerhard referred to it in his talk today. And we can have up to 45-minute apneas, cessation of breathing in this species, and then they'll take anywhere between 30 to 200 breaths, and then you'll get a long apnea again. So these breathing patterns are common, and when the animals are breathing that way, the breathing appears to be quantal. That is, that the apneas are multiples of missed breaths. So that if we make a point care plot where we plot the interval against the preceding interval, if all the breaths are rhythmic, then you get a very tight cluster of breaths. What these other two clusters represent is the last breath before a long apnea and the first breath after a long apnea. And so we see that those come at time intervals that are multiples of the basic rhythm within the episode itself. And here you see in the frog that they can skip 1 breath, 2 breaths, 3 breaths. And so you see multiples of these. So it looks quantal. So again, the questions that were asked is what determines the rhythm when it is expressed, and what turns breathing on and off in these episodes? And this is something that I've been trying to unravel for most of my career. And as it turns out, there's a hierarchy of controls. There's no doubt that feedback from chemoreceptors monitoring, because normally when they breathe, O2 will go up and CO2 will go down, and in the apnea, the CO2 accumulates and the O2 falls. And here we're changing the amount of CO2 that's in gas ventilating, in this case, a frog, and we're also changing the degree of inflation. And with different combinations of stretch and mechanoreceptor feedback and chemoreceptor feedback, we can go from no breathing to single breaths to episodes of breathing and ultimately continuous. So we can produce all the elements of that continuum with the right balance of chemoreceptor and mechanoreceptor feedback. So clearly they play a role. However, we can also take and isolate the brainstem and spinal cord. So there's no feedback coming from chemoreceptors and mechanoreceptors. And in newborn rats and in frogs shown here, we get these beautiful periods of breathing with the long apneas in between. If we take and we transect the brain and take off the pons and all of the thinking bits up front and keep just the caudal medulla, we lose that pattern and the breaths become evenly spaced. And the traces I'm showing here are the raw nerve discharge and the integrated nerve discharge, just so you have an idea what the traces are coming off these preparations. Now, the interesting thing here too is to note that when the breathing becomes evenly spaced, it's slower than the breathing in the episodes, but faster than the apneas, which implies that there has to be a descending excitatory input to produce the tight cluster of breaths and an inhibitory input to produce the long apnea. And these have to be coming, in this case, from somewhere outside the medulla. However, and this just shows the same thing, and we can produce the same effect, not quite so cleanly, by using a high magnesium, low calcium solution on the pontine respiratory group. And if we do that, this blocks synaptic transmission, and if there's no synaptic transmission from the rostral pons to the rostral medulla to the caudal medulla, again, we lose the episodes and we get the more evenly distributed breaths. Finally, if we take these preparations that are only the medulla with single breaths and we cool them and then rewarm them as they start back up, what looks like a single breath when it's expanded is actually a cluster of breaths. So we can get it out of the medulla again alone, but to do so we actually have to cool the medulla to be able to show this. If we take and make a slice that only contains the rhythm generating network, 600 microns wide, we can still record from the 12th cranial nerve that goes to the tongue, which moves with every breath, but there now we can no longer produce episodic breathing. So we've been trying to find out what underlies these descending inputs, and this is a work in progress. So far we found that nitric oxide, GABA-A receptor agonists, mu-opioid receptor agonists, and glycine will produce episodes from single breaths, whereas GABA-B receptor agonists and alpha-1 adenosine receptor agonists will do the opposite. They'll get rid of the episodes and produce evenly distributed breaths. So we know that episodic breathing appears to be fundamental, particularly early in development, but throughout life in many reptiles, amphibians, and fish, not all. The episodes are quantal in nature, and there's a hierarchy of control where we see it in unanesthetized whole animals, anesthetized, in the in situ and in vitro preps, but not in the slice. The unanswered questions, we still don't know the exact source of the excitatory and inhibitory drives. And one of the big questions is, it's so common, but why? What's the biological significance? And that was a question that was asked of Gerhard this morning. We have a number of theories that have everything to do with reducing the mechanical work of breathing by having continuous breaths that will take advantage of elastic recoil and overcoming inertia, which won't be there with single breaths that are evenly spaced, to also reducing O2 levels, which might be important in reducing the production of radical oxygen species, and to enhancing gas exchange, particularly excretion of CO2, which takes more than a single pass of blood through the lung, as opposed to oxygen, which can be loaded very rapidly. But these are all things that still remain unknown. Okay. Moving on, I want to take a little bit of a look now at the afferent inputs, the feedback coming from the respiratory muscles and from the chemoreceptors. And the questions that can be asked here along those lines are, where are they? What do they sense? How sensitive are they? How are they innervated? And what do they do? And this can be done for a whole series of receptors. There are receptors in the airways, in the lungs, both stretch receptors and chemoreceptors within the brainstem, and chemoreceptors within the peripheral arterial arches. And it's of this group, we have worked on all of them, and I'm only going to take a little bit of time and talk about the peripheral arterial chemoreceptors, which is a group that we've been working on extensively lately. Now, most of these are derived from amine precursor uptake and decarboxylation cells, APUD cells, which develop from the neural crest. So as the neural tube forms, neural crest cells migrate, and they migrate to a variety of places throughout the body and are the basis of a lot of sensory neurons and sensory feedback. They produce a whole slew of different neurotransmitters, and they're typically found in terms of chemoreception in the neuroepithelial cells, chromaffin cells, glomus cells. These names get used somewhat interchangeably. They're characterized, the cells, by the presence of a lot of dense cytoplasmic vesicles. Now, it's a picture that I took from the internet of a beautiful set of gills, and you can see the arch and the gill rakers and the pairs of filaments that come off and the lamellae that sit on here. And it's been known for quite some time that the chemoreceptors in fish are distributed throughout the gills. They also occur in some species in the orobranchial cavity, but largely in the gills. And there's a predominance of them in the first gill arch, although for respiratory control, they're found on all gill arches. For many species, for the cardiovascular receptors, all come off the first arch. And back in the 1990s, Pierre Laurent and Suzanne Dunel-Herb and their colleagues noticed that there were glomus-like cells containing serotonin scattered throughout. And we now know that these are found in all species that have been studied. And here we're looking at the filament with the lamellae coming off, and you can see in salmon and trout, they run right up the middle of the filament. In Medaka, the rice fish, they're clustered at the tips. In goldfish, they run up and they tend to be denser towards the tip of the filament, but we also find them out in the lamellae. So there's different types of distributions in different species, but they're definitely there in all. And here we find that they tend to be on the water side more than anywhere else. Oops. Oh, I knew this was going to happen. For whatever it is with a Mac, whenever you turn off and turn on, you lose all your embedded profiles and you have to re-embed them. And unfortunately, that is not going to rotate for us. Whoops. Oh well. We'll have to leave that. At any rate, you can see here that they're sitting on the side where the water coming into the gill, so the afferent side of the gill, they're dense. If this were to rotate, you'd see there are none on the other side. So they're all on one side of the gill. And this work of Michael Johns shows beautifully that these 5-HT containing neuroepithelial cells are innervated by staining with a zebrafish marker for neurons. So you can see the beautiful innervation of the cells. And here in a cross section, you can see them sitting right up against the water and near the efferent filamental artery, where they could sense both water and blood. Michael has done some superb work, and I'm not going to go through the details here, other than to say that their characteristics are identical to those of the carotid body gloma cells in mammals. That is, they have a potassium channel that under hypoxic conditions closes, which leads to the retention of potassium. That potassium depolarizes the cell, activates voltage-gated calcium channels, which lead to exocytosis and release of transmitter from the synaptic vesicles. And it's very much the same in isolated cultured gloma cells as it is in carotid body chemoreceptors from mammals. One of the intriguing things, though, is that we know that they're O2 sensitive and that they release transmitter. Are they the cells that produce the cardiovascular and the respiratory reflexes? And from some earlier studies that we did where we recorded from the nerve coming from isolated gill arches, we could show that there was very little or no response to catecholamines, very small to dopamine, a modest response to serotonin, but a very profound effect to acetylcholine. Now keep in mind, I'm showing you slides where by changing the doses, we could get anything we wanted up there, but these really are looking at close to the maximum responses that we could produce with these drugs. So we've spent a lot of time looking to see if we could find neuroepithelial cells containing acetylcholine and have completely failed to do so. We can stain for 5-HT in trout and goldfish, and we can find gloma cells quite nicely. And we can find bipolar neurons that innervate the gloma cells that contain acetylcholine, but we can't find gloma cells per se. Now, this isn't going to rotate either, I am sure. Oh, it will. No. There we go. Now what we see in this slide is that the blue cells is DAPI staining for cell bodies. The purple is staining for soybean agglutinin, and these are the mucus cells that sit on the surface of the gill. Those mucus cells are rich in acetylcholine. The green cells are the neuroepithelial cells containing serotonin. And as that rotated, let's see if I can do it again, you'll notice that there's a close association between the mucus cells on that side, take my word for it, and the serotonin-containing cells. Well, we also find that if we stain for alpha-bungrotoxin, which is a marker for nicotinic acetylcholine receptors, that we can see a lot of staining, but we find staining to both the serotonin-containing neuroepithelial cells as well as to the soybean agglutinin staining mucus cells. So it appears that both cells have receptors for ACH and that the mucus cells contain ACH. And at this point, in terms of lessons learned, we know that there are abundant neuroepithelial cells that will respond to, now it's been shown by Steve Perry, Chris Wood, as well as Michael Johns, that they respond beautifully to O2, CO2, and to ammonia as a third respiratory gas. And we also find serotonin in mucus secreting cells and Merkel cells, whereas the cells containing ACH, it's only the bipolar neurons, some of the pavement cells, and the mucus cells, and we find receptors on both groups. So in terms of questions unanswered are how these cells are interacting in such a manner that ACH can have such a profound effect while the neuroepithelial cells contain only serotonin. So in terms of modeling how this can go, we've come up with a series of ideas, but this remains to be worked out. Now very quickly, so I don't take a lot of time on this, We find that in all the other species, that the 4 gill arches, which are the 3rd, 4th, 5th, and 6th branchial arch in developing embryos, the 3rd becomes the carotid artery with the internal and external in all of the higher vertebrates. The 4th becomes the aortic arch. The 5th disappears and has no homologue, and the 6th becomes the pulmonary artery going to the lungs. And so these are the homologies of the arches, and what we find, if we take a look, is that then our chemoreceptors go from being in the gills in the fish to being in the carotid labyrinth, a very elaborate structure in amphibians, which receives an arterial common carotid artery comes in. It breaks up into this massive sinusoid that then amalgamates to form the internal and external carotid arteries, and the receptors sit within there. We find that once you can finally find the area where the receptor cells are in reptiles, if you open the artery up, it's a honeycomb and a lattice with all of these struts and cross bridges, and the chemoreceptor cells sit within the struts and the cross bridges themselves. And by the time we get up into mammals, the carotid body sits as an independent organ on the surface right at the carotid bifurcation. So we see this evolutionary trend in the structure. We find then, along with that, the innervation goes from being by both the 9th and 10th cranial nerves. In mammals, it's only the 9th. In reptiles, it's a mishmash of either 9 and 10 together or 10 alone and 10 alone in birds. And what we see in terms of neurotransmitters is 5-HT and ACH in this arrangement that I've discussed. By the time we get to the gloma cells, you've got acetylcholine, serotonin, dopamine, norepinephrine, substance P, bradykinin. You've got a plethora of transmitters all in the same cell. And in between, we find in the amphibians, no acetylcholine, just catecholamines and 5-HT. And in the reptiles, ACH and 5-HT, but always in separate cells. The unanswered questions, and the big one to us, are what are the relative roles of these different receptors, particularly the ones in different locations. We have a theory, has a little bit of data to support it, and that is the receptors on the first gill arch and the ones that become the carotid body and the carotid labyrinth are more involved in controlling breathing, and that's oxygen supply, picking it up from the atmosphere and delivering it to the tissues, and that the ones associated with the second arch that become the aortic arch chemoreceptors are more associated with sensing content rather than partial pressure, and leading to, if the content is low, erythropoiesis and the production of more red blood cells, increased blood volume, so there's more carrying capacity, and that they take on different roles evolutionarily. Okay, finally, the one last thing I want to do on this pass-through is talk about the interactions that occur over on this half of the control diagram. Now, one of the things we know, a feedback loop is a loop. Anything that's a circle, you can enter and leave at any point. So right now, if the oxygen were to fall in this room, it would stimulate the chemoreceptors and breathing will go up. But by the same token, if you start to breathe hard just sitting there, that increased ventilation will enhance the oxygen levels, which ultimately will lead to the controller trying to turn the breathing down. So breathing can change blood gases can change breathing. And notice in this diagram we have that same interaction between the lung gases and the blood gases and metabolic rate. And the key message here is that feedback loops are circular, and anything can be cause or effect. And trying to sort out cause from effect can be difficult. And I'm going to give you one example. So here we see the ventilatory responses, and this is for a ground squirrel, 2 species of ground squirrels, but we drop the O2 and breathing goes up. A key thing to keep in mind is that there are time domains to ventilatory responses, and this is something that we've worked out well in mammals, but not so well in other species. But if you look here now, when we expose this animal to 7% oxygen, we see a rapid rise in breathing that then falls off over the next half hour and then begins to go up again over time. Now, what this reflects is a series of underlying changes that are going on throughout that central control system. And for mammals now, we have worked out a whole host of time domains within the response, some of them taking place within seconds, some of them taking place over a lifetime. When you measure the oxygen, the hypoxic response is important. If you don't want to be comparing apples with oranges, you need to be comparing the same components. Compare acute response to acute response, or compare different parts of the time domain with one another. Well, we find that the same thing is true in what we call the hypoxic metabolic response. If we expose, again, ground squirrels to 7% oxygen, we see over time the metabolic rate drops, as shown here by a drop in body temperature. Over here, it's metabolic rate itself, and again, you can see time domains where we see an increase, we see it rolling off, and if we were to go out for 2 days, you would see that this returns all the way back up to normal again. So once again, there are time domains to the metabolic response that have to be taken into account. But when we look at this metabolic response that we see here, this fall in metabolism over time, is that due to the fact that the ventilatory response is not delivering sufficient oxygen, and therefore the animals can't maintain metabolism, or something else? And with Glenn Tattersall, we addressed this question, and what we did is we began by heating and cooling only the hypothalamus. And if we do that, you see a very narrow range where the temperature of these animals is about 39 degrees. And if we heat or cool, you begin to see metabolic rate go up as the animals either try to cool or try to warm themselves or try to cool themselves. If we make them hypoxic, you see metabolic rate comes down. You see that the set point for body temperature regulation is shifted to a lower temperature, and there's a blunted response, metabolic response, to further heating and cooling. So that it's the set point for body temperature that's going down. There's a decrease in heat production. To get there, they actually dump heat, so that here we were recording with an infrared camera temperature at the windows for heat loss within the animal, and you can see over time they open up, they vasodilate, they lose heat, and as they come down to the new set point, they shut down again and become stable. And during that period, shivering is completely inhibited. So while they're on the low oxygen, we see at 25 degrees that there's no shivering at all. And then if we take them down, here breathing air, you see intense shivering, which is absence in a hypoxia, until the moment you take the hypoxia off and the shivering becomes expressed. So it's a very orchestrated suite of events that brings, when oxygen is limiting, brings metabolic rate down. So not only do you get a ventilatory response that increases supply, you get the metabolic response that reduces demand. Okay, so the greater the depression, the smaller the response. The greater the response, the smaller the depression. Cause and effect again, hard to sort out. So the lessons learned is there's a hierarchy again here, that it's a fundamental process, it occurs in all animals to different degrees, and that there's a hierarchy of control. And the big question is, how are these processes integrated? Okay, finally then, I've been looking at individual components. Another big part of my research has been trying to put it all together and look at how plastic these processes are in allowing animals to adapt to environmental extremes. We've done a lot of work on the low end on animals in sleep, in torpor, in hibernation, and what happens when you turn down the need for oxygen. We've done a lot of work with diving species where we have a combination of the need to reduce the rate at which oxygen is used to prolong dive time in animals that are actually exercising during those breath hold and those apneas. And what I want to talk a little bit about tonight is my one example from all the sorts of things we've done is looking at what happens in birds in flight. A number of studies looking at entrainment of breathing with wing beat during flight, as well as the new work that we've been doing looking at flight at altitude. Now, The species we've focused on is the Barheaded goose, and the reason we've chosen that is these birds spend their winter getting fat in India, where most of the Indian subcontinent is not much above sea level, but to reproduce they fly over the Himalayas up into Chinese Tibet or up into Mongolia, crossing over the highest mountain range on Earth. The questions we ask are, how high do they fly? How energetically demanding is it? And how do they match supply and demand under these conditions? Well, again, we come back to the transport cascade. And if I go through it quickly, because there's only one key point I want to make, in terms of ventilation, we see by comparing Barheaded geese to other species that the breathing pattern they use has much higher tidal volumes and lower breathing frequencies than we see in other birds. They have a very vigorous response, so that they breathe hard with a pattern that is more effective in terms of gas exchange with less dead space ventilation. In terms of pulmonary diffusion, we find that relative to other species of geese, they have much larger lungs. We've been working with John West. We had a hunch that they might have smaller air spaces and a smaller diffusion distance between the airspace and the red blood cells across the endothelium and epithelium. It's turning out that that's not the case. We don't have final data yet, but it doesn't look like the barrier is any thicker or that the parabronchial spaces are any smaller, but there is no doubt that they do have more surface area due to larger lungs. On the perfusion side, compared to barnacle geese shown in blue, the Barheaded geese have a left-shifted oxygen equilibrium curve, and as a result, we see that at any given arterial level, they are carrying a third more oxygen in the blood than the barnacle geese, a close sister group. And we find at the tissue diffusion side a whole host of differences, more oxidative fibers. We find that the mitochondrial volume density for given muscles tend to be higher, and that there are more capillaries relative to the mitochondrial density. We find that more of the mitochondria sit in subsarcolemmal locations, and so that there are a number of adaptations that enhance diffusion of the oxygen, which is left shifted out of the blood and into the tissue itself. So, bottom answer question here is, and not surprising, within the cascade we see adaptations at every single level that are going to enhance that flow. Now, questions that come up, since all of this is done in domestic Barheaded geese born and raised at sea level, under resting conditions, what happens in birds in the wild and what happens to birds in flight? And this is work that we've been addressing with a series of studies. First of all, we've gone to Chinese Tibet and worked on wild barheads that are, oops, a little dyslexia there, wild bird barheads that are born at 3,200, 3,300 meters. Some of them are born up to 5,000 meters. The population we work on, about 33. A group that have been in captivity at altitude for a year or more and therefore have not been exercising, are sea level barheads and barnacle geese. And again, you see that if we take a look at a partial pressure of about 40 millimeters mercury, that again, our sea level are doing better than the barnacle geese, but the wild geese are doing even better in terms of their ability to load oxygen. And we're seeing a variety of adaptations in the wild population that we don't see in the domestic population. So yes, there are differences, but this is an ongoing study. Sabine Legou will present some of this at the animal biology session tomorrow. So give her a plug to go there. We've also been looking at birds in flight in a wind tunnel at UBC, and we'll see if I, get lucky on this one or not. I do. And so here in slow motion, you can see our birds flying. They've been trained. They were imprinted at birth on my graduate students and postdocs. Jessica Meyer is the one primarily involved with this, along with Julia York and Michelle Reichert. And through the mask, you can see two tubes. One is drawing air to measure metabolism. The other is providing nitrogen so we can make them hypoxic, simulating altitudes of various levels. On the back, they have a backpack that's carrying a device that measures blood temperature and heart rate. And so we're beginning to get data from flying birds at simulated altitude. But one of the key parts of this is the work that we've been doing in birds from Mongolia. And the Mongolian birds make that cross over the Tibetan Plateau, over the Himalaya, and down into India and back. Here's a profile of the route, and you can see India is indeed flat, and then you hit the wall of the Himalaya, which the birds need to cross and get over to the breeding grounds in Mongolia. Now, interestingly, out of the early studies we've done, you see that here, these are the flight profiles for a number of birds, and you can see that they leave India at sea level, and they are up to 6,000 meters, 5,000 to 6,000 meters, and through the pass in under 12 hours. So no acclimatization, rapid ascent over the top in half a day. They're doing it during the night where they're not taking advantage of the updrafts. So the arrows here show the direction of the wind and the length shows the speed, but they're doing it when there's a mild downdraft. Now, well, I'll mention again in a moment, and that they're typically flying close to the ground. Our GPS is not the most accurate, so you can see based on where we know the locations of the birds, they appear to be burrowing at times, but they're not really. They are flying, but they're flying low to the ground. Now, most recently, and Charles Bishop presented some of this data earlier today, and this is from data loggers that we've had out over the last few years. And here are profiles of birds now flying from Mongolia down to India. And these are different birds on different days, but here's one just leaving Mongolia at different points in the Himalayan crossing and descending into India. And with Charles's loggers, we've been able to record heart rate, acceleration, using 3D accelerometry, and from that we can derive wing beat frequency. And what this data shows is that on occasion the birds do get favorable winds that they take advantage of, but for the most part it's continuous flapping flight. And out of this, Charles has shown a beautiful correlation between heart rate and wing beat frequency. And with the work in the wind tunnel, we've been looking at the correlation between heart rate and wing beat frequency, so that if luck will have it, we can come back and answer the question of how energetically demanding it is by taking the wing beat and the heart rate data and using that to derive oxygen consumption. In terms of flight, the birds aren't dumb. They go through a pass if they can, and so they seem to be crossing in the 5,500 to 6,000 meter range. The highest we've recorded is 7,200 meters, I believe. Can they fly higher? That's something that we argue amongst ourselves, but right now the data say typically they'll go through a pass if they can go through a pass. How do they match supply and demand? They do it at all levels of the cascade. Okay, so now the fun in life is looking at this plasticity and looking at various animals adapted to environmental extremes across the full spectrum. And that's been a great part of my career, is looking at these different ones. I've given you one example tonight. And on that, I want to do two things. First is an acknowledgement, one to NSERC, which has funded my entire career, and it's one of the few agencies that will allow me to dabble as widely as I do and do the things I can. There aren't too many funding agencies that will allow you to be as unfocused as I have been throughout my career. I need to acknowledge my wife, who tolerates me, and without her support, I wouldn't be able to do these. As long as there's a cup of coffee in her hand, I can take her, or she can take me just about anywhere. I want to acknowledge specifically the group that's been involved in the Wild Barheaded Goose Project. Graham was really the motivation to get this started, along with Fraps, and then we approached Pat and Charles, and they really carried the ball from that point on, and have been the driving force behind this, along with Lucy Hawkes, a postdoc, now a faculty member at Exeter, and Bev Chua, who's my clinical vet, who does all the surgeries in the field for me. And I also want to acknowledge now my graduate students, postdocs, and research associates over the years. I have, If you look at any thermal images or images of me, you'll notice I have a pinhead. It's the smallest of any in my group. I have one pair of hands and a very small brain. And most of this work reflects the fact that I've been lucky enough to recruit people with very good hands and much larger brains. And it's to them that I owe so much of the success that I've had. And this is primarily the group, many of whom are in the audience, many of whom you may recognize from earlier and younger days. And my dog. Now, let me do one last thing for that. I have one last thing, which is a request that's come from those who didn't attend the party they had for me in my retirement last year. And I'll see if I can pull it off a second time. I don't know if I can.
Well, this is blow you away.
I am a comparative physiologist, a respiratory neurobiologist, an eco-biomedico schizophrenic that has an approach that is multicultural, postmodern, reductionist, and politically and anatomically and physiologically incorrect. I've published data without stats, stats without data, reviews without data, data that come to no conclusion. I am consistently inconsistent. I embrace the 5 sins of comparative physiology: symorphosis, scaling, safety factors, strong eye indifference, and systematics without believing in a single one. I'm a non-believer, an overachiever. I'm laid back but fashion forward. I'm upfront, down home, low rent but high maintenance. I study animal respiration, graduate student desperation. I'm a listener, a motivator, but most of all, I knew I was going to lose it, but most of all, a delegator. I cannulate, I cauterize, I ablate, I denervate. I have a reputation I haven't earned. I have incredible luck and many lessons learned. I am an aging child, prematurely post-traumatic. Prematurely post-traumatic. Oh, come to me now. I'm approaching retirement. I'll jump a line. I'm approaching retirement, but not giving up. I'll take it slow, go with the flow, ride with the tide, keep glide in my stride. But there's one thing, there is no doubt, I'm not giving up and I'm not yet out. Thank you.